The goal of our research was to identify possible orthologs between Meiothermus ruber and E. coli. The results obtained from this research was that MRUB0320 gene is an ortholog with B3452. The MRUB0321 gene is an ortholog with the B3451. The third pair was that MRUB0322 gene is an ortholog with B3453. And lastly, the MRUB2366 is an ortholog with B3450. These are all found in E. coli, which encodes for components of the ABC transporter specifically involved in the SN-glycerol 3-phosphate. This research was done by me, Jenna Hall, and Joel Nelson in the winter of 2017 to 2018. Specifically, we are using Meiothermus ruber. The reason we are using it is because it's a study organism. Specifically, some characteristics about Meiothermus ruber is that it's a gram-negative rod-forming bacteria, as can be seen by the images below. It's also a thermophile, which means it grows from 35 to 70 degrees Celsius, most efficiently at 60 degrees Celsius. It also has a red pigment. The reason we're using it as a model organism is because it hasn't been studied extensively yet. Using Meiothermus ruber as a study model organism has many benefits, which include improved identification of orthologs, which is our goal, improved phylogenetic anchoring, gene discovery, and classification of other microbes. Specifically, we're focusing on the glycerol 3-phosphate of these ABC transporters. So ABC transporters are very important because they're crucial to all organisms. Transport systems bring external nutrients and eliminate harmful toxins. These transporters are both importers and exporters. Transporters are diverse since they must be used to achieve many different tasks. These ABC transporters have ATP binding cassettes, which is where they get their ABC name. The ATP binding domains with the presence of a phosphate binding loop and a short consensus sequence of LSGGQ. These ABC transporters are increasingly significant because they make up the largest protein family. Specifically, we're talking about an ABC transport family of glycerol 3-phosphate, which actually participates in glycolysis, gluconeogenesis, lipid synthesis, and electron transfers into the mitochondria. Since these are highly important in the metabolic regulation within a cell, it is increasingly important to do research on this. Next, I'll talk about the function of each of these proteins that we're going to be looking at. So the pathway that's shown is the pathway of the glycerol 3-phosphate transporters actually in E. coli as an example. You can see that the genes shown, the UGP B, A, E, and C, are all part of an operon, which actually transcribes to be able to get the proteins at the end, which is the UGP E, C, B, and A. The E. coli we're using is a K12MG1655 locus tag. It's involved in the glycerol 3-phosphate transport of B3450, B3451, 3452, and 3453. Thus, looking through this, you can see further when I indicate a B in front of the gene that it's an E. coli gene that we are talking about. So the genes that are in this all have a different function, but all combine together to involve in the ABC transport. So the first gene that I'm going to be looking at is the UGPA, which represents the B3452 in E. coli, and the MRUB0320 and M. ruber. These both transcribe to have an ABC transport permease protein. The UGPB gene, which is the B3453 in E. coli, and in M. ruber, it's the 0322, which functions as the ABC transport periplasmic binding protein. The third one is the UGPC, which is the B3450, which functions as the ABC transport ATPase. The last one is the UGPE, which is the B3451 in E. coli, and in M. ruber, it's the 0321. This functions as the ABC transporter permease as well. So as you can see, all of these transcribe for proteins that play a role in ABC transport.
To begin the analysis of these sets of genes in the SN-glycerol 3-phosphate ABC transporter pathway, multiple databases were used. Through the use of KEGG, the genes of interest were found for both M. ruber and E. coli. In the figure seen here, the E. coli genes are located on the left side, while the M. ruber genes are found on the right. A slight variation in this specific analysis comes from the fact that mRub2366 does not seem to be part of the SN-glycerol 3-phosphate specifically, as seen in this figure. When we originally started comparing these genes, you can notice that the UGPC is not found within the M. ruber genome. When a blast with B3450 was performed to identify what possible genes could be a part of the system, mRub2366 was found. Through investigating this further, mRub2366 was found to be the ATP binding domain for many different monosaccharides, including SN-glycerol 3-phosphate. This is somewhat of a variation in the fact that this entire system of the SN-glycerol 3-phosphate ABC transporter is not a full operon in the case of the Meiothermus ruber system. We use a variety of bioinformatics tools in order to gain a better understanding of all of our genes of interest, and I will briefly discuss the use of each of the ones that we used personally. TM-HMM use the amino acid sequence of the gene in order to predict the possible number of transmembrane helices. This helps identify if it is a transmembrane protein or if it's located elsewhere in the cell. Signal P use the amino acid sequence of the protein of interest in order to predict the probability of it containing a signal peptide. LipoP similarly use the amino acid sequence of the protein of interest in order to predict the probability of the protein it creates having either a lipoprotein or a signal peptide within its structure. In both of these cases, a signal peptide or lipoprotein presence would help narrow down the location of the protein of interest. P-sort B used the amino acid sequence of the protein of interest and calculated probabilities of the overall location, whether it would be in the cytoplasm, cytoplasmic membrane, cell wall, periplasm, outer membrane, or in the extracellular space. We found this tool to be very helpful in identifying the location specifically. Phobius was the last cell localization tool used, and it helped us identify whether the protein of interest was found in the cytoplasmic or non-cytoplasmic space. This tool was a great way to help confirm further predictions on cellular location of the protein. Now moving on to the protein family comparison, the CDD, or Conserved Domain Database, used the amino acid sequence of the gene of interest to identify a COG number that indicates whether two genes are orthologous. Along with this output is an e-value that indicates how strong of a correlation it is. PFAM used the amino acid sequence of the gene of interest to identify a PFAM number that indicates, again, whether the two genes are orthologous. Along with its output is another e-value that indicates how strong this correlation is. TigerFAM was yet another one that used the amino acid sequence of the gene of interest to identify a TigerFAM number this time to indicate whether the two genes are orthologous. Again, an e-value was used to indicate how strong that correlation is. Lastly, PDB, or Protein Data Bank, was used by inserting the amino acid sequence and trying to identify crystalline structures that were used with another e-value for comparison. Through the IMGM database and within the color by KEGG function of this site, genes of interest could be found within the specific genome, and the location and function of said genes could be compared. With this, color by KEGG coordinates functions of genes with specific colors. The three genes of interest are indicated by the red lines found above the gene. In this case, from the top, mRub2366, mRub0321, and B3450. The top two panels is the M. ruber genes, and the bottom is the E. coli, which are colored purple and green respectively. Both of these colors indicate that they are involved in membrane transport. You can see the comparison of whether an operon is present as well. With the M. ruber genome, mRub2366 is clearly found far off from the rest of the operon, so that portion is not included in the SN-glycerol 3-phosphate ABC transporter operon, while the entire E. coli operon can clearly be seen located altogether.
The first pair of genes that were analyzed, we put together a table which you'll see for each of the genes. This table indicates on the left side of it is the B gene, which is going to be the E. coli, and the right side will be the Meiothermus ruber. In this case, I'm showing support for B3450 and mRub2366 being orthologous to each other. Numerous bioinformatics tools were used to support that these two genes are orthologous. The first task to be done is to analyze the blast nucleotide sequence between mRub2366 against B3450. The blast results gave a low e-value, indicating that these genes are highly correlated, which is not due to chance. These genes also have the same COG number, as you can see on the chart, and TigerFAM number and PFAM number, which is another piece of evidence proving that these are all orthologous. The last piece of evidence is that that these two genes have the same cellular localization. The cellular localization is identified as being embedded in the inner plasma membrane, which is supported through TMHMM, signal P, lipo P, PSORB, and phobias, which we've talked about before about all of the different methods that we use to support this. For both E. coli and mRub systems, the genes found were in the ABC transporter page of the KEGG map, giving another reason that they are orthologous. The only difference in the data found was that the protein database where the two genes represent actually different proteins, which could be due to an incomplete database or that there might be altered proteins in these databases that may actually be the same, but because they are from different species, they may represent a different name. This piece of evidence can be disregarded because there's a significant number of evidence gathered that confirms that mRub 2366 and B3450 are orthologs. The second pair that we analyzed was the E. coli gene B3451 and the mRuber gene mRub0321. These also were shown to be orthologous. So the process is the same between these two genes. These represent the UGP and the E in the respective genomes. This table, again, we go through, show the BLAST, which also has low E values, again, showing that they are orthologous. The COG and PFAM numbers are the same in the both genes. The cellular localization, just as the previous one, shown by TMHMM, signal P, lipo P, PSORB, and phobias are all similar, showing that they have the same localization in the plasma membrane. The protein database in this one actually does show that they are the same and the KEGG map, again, is the same, that they are found on the ABC transporter page. The only piece of evidence that differs between these two genes was the TigerFAM database, which did not give the same hits as the B3451, did not yield any hits. This provides an inconclusive data set, which does not exclude that they aren't orthologous. In fact, this is another piece of evidence that overall we can conclude that B3451 and mRub0321 are actually orthologous with respect to the rest of the supporting data that we have found.
The third gene pair analyzed was mRub0320 and B3452, both of which are classified as a UGPA in their respective genomes. Again, for ease of understanding, this table is used to compare the bioinformatics tools. The BLAST performed compared these genes, produced a low E value, again, indicating the genes are orthologous. The COG number identified through the CDD database was the same, with slightly varied E values. Using the cellular localization data mentioned before, both genes were confirmed to have the same location. The TigerFAM numbers had varied results between the two genes, but with two different TigerFAM numbers showing up. To continue, PFAM database showed the same protein family between the two genes with similar E values. The protein databases showed different proteins of similarities, but for both of these systems, the genes were found within the ABC transporter KEGG map. Nearly all forms of data helped conclude that B3452 and 0320 of mRub are orthologous. Regardless of the TigerFAM and PFAM results, this connection is strong. The last gene pair analyzed was mRub0322 and B3453, both of which are classified as UGP in their respective genomes. We have, again, another table to compare the bioinformatic tools used. The BLAST database, again, gives low E values indicating that they're orthologous. The COG number identified was the same for both genes, as well as the cellular localization data showed that both genes were confirmed to have the same location. PFAM database shows the same protein family between the two genes with very similar E values, while neither gene had a hit with the TigerFAM. No hit in TigerFAM leads to inconclusive results because the several other pieces of evidence are strong that it can be concluded that mRub0322 and B3453 are orthologous to each other.
Throughout all the bioinformatic tools, it can be concluded that mRub0320 and B3452 are orthologous, as well as the rest of the other pair of orthologs. There are instances of differences between the ortholog pairs, but the majority of evidence has been clearly shown that they have distinct similarities between each pair. Since the two organisms are distantly related, they may have contributed to the slight differences in analysis. However, the overall data has supported that all of the pairs that we have shown are orthologous to one another. All of these genes are also a part of the operons that do involve membrane transport, as shown in the figures above by the color by KEGG database. The ABC transporters found in are found in almost all organisms, making them universal. Makes this study increasingly important. The research shown can be linked to ABC transporter systems involved in glycerol 3-phosphate. These transport systems are linked to diabetes, cancers, and other metabolic diseases.